Bueno, muy buenas tardes a todos. Gracias por la puntualidad, los que llegaron puntuales, y esperemos que los demás se incorporen. Por respeto a los que están en línea, ya vamos a comenzar. El día de hoy sí tenemos el evento, este curso, de 2 a 4, pero el día de mañana, por favor anótenlo, va a ser de 3 a 5 por problemas logísticos del auditorio. Y la próxima semana les vamos a ir avisando si logramos que todo el curso sea aquí en el auditorio, y si no tendrá que ser el cambio como lo hemos estado haciendo aquí en las aulas del 18. Pero les avisamos todos los días cómo va a estar para que no se hagan bolas.
El viernes.
Sí, mañana viernes.
No, mañana es jueves.
Perdón. Sí, ¿verdad? Aquí Constantino está poniendo nerviosa. Bueno, mañana, ya les había dicho, va a ser el curso de 3 a 5 y el viernes otra vez de 2 a 4. Y ya veremos, ya les iremos informando los demás días cómo van a quedar. Bueno, pues muchas gracias, le damos las gracias al doctor Cristian y pues por favor, comience el curso. Gracias.
Okay, thank you very much for this introduction again, and yeah, we have to move a bit forward. Yeah, that's it. Thank you. So I was told that I was last time not close enough to the microphone. I hope it's better now. I will try to be a bit closer. Yeah, so let's catch up where we stopped yesterday. What did we learn yesterday? Yesterday we talked about photosynthesis. So you got the process of the electron transport chain in order to perform the light reaction photosynthesis. We also talked about the Calvin cycle, which is the dark part of photosynthesis. And this was basically a short introduction in biochemical terms. We will come back to this today. So today you will hear about another electron transport chain. So if you paid good attention yesterday, then I think it will be really easy to follow today. It's maybe a bit difficult for the ones that missed the lecture last time yesterday because we had no online option, but I will try my best to make it as clear as possible so that everyone can follow. Well, after this introduction, we started to talk about microbiomes. So we got an idea what a microbiome is, and we also tried to differentiate it from the term biocenosis. And we also talked about possibilities to analyze microbiomes. First, we talked a little bit about microscopy. Here you also had some ideas how to apply microscopy, what could work, what could not work, and we discussed the limitations of microscopy. And from this we came to the sequencing, which allows us to analyze much more organisms in parallel than just based on microscopy. Well, based on this, we started then to talk about DNA and about the possibility to perform this polymer chain reaction in order to amplify the so-called 16S rRNA gene. It's this one specific gene that we like to use in order to compare different organisms, different microorganisms, in order to find out who they are. Yeah, and in this regard, we also saw a short video with the nanopore technology. I can shortly go back. This was the machine that I showed to you, where we can basically drop in our amplified DNA, and there we can sequence then our DNA. We saw this video, sorry. We saw this video with these porins. So there was like a membrane, and in this membrane we have molecular pores, and we are measuring the current across these pores. So there are some proteins running through, some electrons running through, and then we have really small pH meters to measure the current there. And so we can basically measure millions of sequences at a time in such a device. And in this video, it might be a bit confusing yesterday because you only saw small structures that were going into this pore. And you can do actually the same with DNA. So we have these small pores. The DNA goes through this pore, and with every nucleotide, the current changes, which is going through the pore. And then we can measure the current, and then we actually can say which nucleotide was measured at a respective measurement point. Yeah, this was just a short summary, and now we are at a state where we can read our DNA. So we put it in our sequencer, and then we get these sequences. This is a very short one, and they can be much longer, for example, like this. This is one of these 16S rRNA genes. It's from an organism called Prevotella pallens, and I just took it by accident. I just searched for any 16S sequences. We could also show other 16S sequences. So what can we do now with this? What would you do with this? How can we come from this to an organism? Anyone has an idea? No one has an idea? Maybe you? Yeah? You have no idea?
Could you repeat the question, please?
Yes, sure. So we learned yesterday about how to perform sequencing, which we need to analyze lots of microbes to find out who they are. And now we sequenced this gene, just as one exemplary gene. It's one 16S gene from this organism. If we don't know that this is the organism, if we, let's say, have an unknown microorganism, we sequence the 16S rRNA gene, we get this. How do we come from this to the name? How can we find out to which organism the sequence belongs? You have an idea?
Yes, I think that we could make a database for that sequence for a lot of organism and just search what that sequence for what microorganisms belong.
That's exactly right. Thank you. That's how we should do it. Yeah, so what can we do with this sequence? So we could, for example, use DNA to make structural analysis, identification of genes, and functional analysis. That's what we mentioned already yesterday, but that's what we can't do with this 16S rRNA gene. For this, we would have to sequence all the DNA. But we learned this is quite expensive, and we want to reduce the effort. We want to save some money. So we only sequence this one 16S rRNA gene. And with this, we can make some conclusions about phylogenetics. We can measure how close different sequences are to each other. And this is also the basis for classification. So if we have a database, as you suggested, and we compare the different 16S genes, and they are extremely similar, then we know that's our organism. Yeah, how does this work? How can we actually compare the sequences with each other? It's maybe not that easy. There's a nice tool that you can use online. It's actually for free. You can just go on Google and you look for the Clustal online platform. And then you can just by copy and paste take your 16S gene and then you put it into the cluster. And then you take a second one where you know which organism this is, and then the system automatically starts to compare the sequences. Here, for example, you have a part of the sequence which is not similar to the other one. Then you have some similarities which are marked with a star. Sometimes the algorithm includes some gaps on its own in order to make it fit better with later sequences, which then fit good together. And like this, the algorithm is going through the whole gene, and then in the end you can count these stars and calculate a similarity value based on this. So this is quite time consuming if I do this manually, but there are tools that we can use to automatize this. So there's a program, for example, called KIME, and then you can feed in all your sequences, millions of sequences, and automatically KIME is comparing every sequence in this manner to the sequences in your database. And like this, in the end, you get a list of all the organisms that you found. And with this, you can create a taxonomic profile. Oh, that's still German, but it explains what I just explained to you. So we can use it either to identify organisms, but we can also create taxonomic trees. We already talked about the tree of life. You may remember that's actually the first tree of life that was developed. This was already quite nice. It was from a guy called Lamarck in 1809. And he was doing this just based on phylogenetics, on the phenotype. So he was just comparing the organisms, whether they look similar, whether they have the same amount of legs or arms or whatever, whether they have hair or fur. So all these kinds of things he was comparing, and then he could make this tree based on a visual comparison of similarity. But that's not how we approach this today. There was quite a development, and the next example was from Haeckel. He was trying to build the tree further, and then he came up with a tree where man was at the top, so like the top of evolution, but that's now a theory that we also deny. And all changed with Watson and Crick. I think you know them, you have heard of them. Those are the guys which actually came up with the structure of the DNA. This was when DNA research started, and shortly afterwards we were able to sequence DNA, and with this we could now build up much better trees like this one. So this already looks quite complicated. It's a recent one from 2016. They always update them, so they're never ready. There are so many organisms which we still don't know. So it's a continuous process, and those are just major groups. So you can see here already big bunches of lines. Every line shows one group of organism, and theoretically you could go further if you go to genus level or species level, so to more accurate classifications, then this tree would explode in a complexity that we can't show anymore. There are trials to still create such a complex tree of life. There's a webpage that you can use, for example, from NCBI. I don't know whether you have heard of this. This is the National Center for Bioinformatics in USA, and they provide this website for free. So you can just enter NCBI, and then at somewhere in this webpage you have like a text-based tree of life. They don't visualize it anymore, but then you can find all the organisms that we have so far. Well, this was just for completeness. I'm already happy if you remember this for the lecture. We just need it very simple. So we need to know that we have bacteria, archaea, and eukaryotes. All of them are derived from the lowest universal common ancestor. And we have much more bacteria than anything else. Not only in number, but also in terms of biomass. So with this, we would probably not say anymore that mankind is ruling the world, but it's probably much more accurate to say that bacteria are ruling our world. So how does DNA sequencing now work? I explained most of the processes that we need to create like a very simple protocol. So how would you start? How would you perform DNA sequencing? Which steps would you do? Try to be creative. It doesn't need to be right. I'm just curious what we think about this procedure. Where would you start? What would be the first step that we have to do?
It's that process that start with hit ¿cómo se dice muestra? Sample, the sample at 19.5 Celsius degree.
That's the polymerase chain reaction. Yes. That's the DNA amplification. Well, that's one important step. That's right. But we need something before.
Okay, no, sorry.
Before we amplify, what do we need? It's a process that I didn't explain yesterday, but maybe you figure it out.
Extract the DNA?
Who was saying this? Ah, yeah, that's right. We need to extract the DNA. So we extract the DNA, then we amplify it, and then what? Transcription, no. I think I know what you want to say, but the term is not right. Yes. Yeah, to amplify it, yes. Read it, yeah. And we're doing this with... Yeah, with a sequencing device like the nanopore. Pardon? Yeah. Well, the reading is done in this device that we saw before. Here, and this is basically a membrane with a little pore inside, and then the DNA is going through the pore. And with every nucleotide passing the pore, the current changes, and then we have like a diagram with fluctuating current, and this can be related to the DNA sequence in the end. Well, and then, what's the last step? You said it?
Compare it to the database?
That's right. So, that's now all the steps together. So now we know how to read the DNA. The database that you were mentioning is actually called Silva or Green Genes. Those are like two never-ending projects where the responsible people are trying to collect more and more 16S genes. Daily, more genes are discovered, which are then added to these databases. It's actually just a file that you can download. And then you can implement this file in your KIME program in order to compare it to the sequences that we just produced with our sequencer. And now we have a list of organisms that we can watch in Excel, for example. We can make a diagram which just shows the taxonomic profiles. So there are other techniques than DNA sequencing. Maybe you remember that I introduced the term omics. So anyone knows what omics means? So, yeah, it's a really common term in bioinformatics especially. And we use it to describe that we want to analyze an entity of many things which are really similar to each other. For example, we can analyze all DNA. And DNA molecules, although the sequence is different, on a chemical way, the DNA behaves always similar. So we have many different, many similar molecules that we are analyzing, and this describes omics. And we can do this not just with DNA, but we can also do it with, let's say, epigenomics. Do you have here the term epigenomics? So you remember that DNA stores all the information from our body, from all the things that our cells can do. But all the information is not always used. And the cell can actually modify this by epigenomics. So there can be proteins which are blocking certain genes so that they can't be read by the polymerase. Or we can have a condensed region where the DNA just aggregates together and then can't be accessed anymore by any proteins. So there are many ways to modulate the DNA in order to restrict certain information and to make sure that only certain informations are read from the cell. So this is called epigenomics, and this can also be analyzed. Or transcriptomics. Transcriptomics is related to the RNA. The RNA, I guess you still remember. Or we can analyze the proteome, performing proteomics. And the proteome contains all the proteins that we are interested in. And then we have the metabolome, which is about all kinds of metabolites that we want to analyze. So those are quite some methods. We could also apply all of them, and then we are doing panomics. But this would already be quite expensive. For the moment, we restrict to DNA. We will have a separate lecture where we're just talking about taxonomic profiles based on genomics. And then we will have another lecture, if the time suffices, where we will talk a little bit about proteomics. And then I would say the other omics approaches would just be too much in the frame of this lecture. Okay, at this point, I would give you a very short summary. So again, we introduced the term microbiome. This was still done yesterday. We saw that microbiomes contain single-celled algae, fungi, prokaryotes, which contain bacteria and archaea. And all of these can be found in a microbiome, which we can also refer to as a biosynosis, although a biosynosis is much more than a microbiome, because a biosynosis can also contain real animals, everything that lives and thrives in a certain space. Yeah, then we introduced the 16S RNA gene, which we need to compare different organisms in order to build our taxonomic profiles, and we learned the methodologies that we could apply to perform this analysis. DNA extraction, DNA amplification, DNA sequencing, and then the analysis based on databases. And finally, we heard about the omics approach in regard to the analysis of large entities of similar molecules like DNA or proteins. Okay, I hope this is clear. Are there any questions? Okay, great. Then we move forward, and now we're going to talk a bit more about specific microbiomes. Now we heard very generally about the microbiome term, but there are different microbiomes that we can distinguish. And here are just some examples. I included some articles which are focused on these respective microbiomes. If you're interested in this, you can read them, but it's not further relevant for the lecture. So we can, for example, find a microbiome in indoor environments like in a sauna or in a refrigerator. We can find microbiomes in anaerobic digestion or in wastewater treatment. We can also find microbiomes in soil. Basic everywhere. So everywhere where we look at, even in the air, we have microorganisms. So we could also analyze the air microbiome. But of course, in the course of this lecture, we will only focus on anaerobic digestion, and we will also talk a little bit about wastewater treatment. There's an interesting term that you might have heard about called microbiome engineering. It's about the idea to change the microbiome specifically, to change its functionality or to make it more efficient. And there's an interesting article, a mini review, which was published 2017. It's not too long ago. So all this topic of microbiome analysis and microbiome engineering is a relatively young topic of research. And I included here some interesting citations from this review article. For example, the analysis of microbiomes is very often about culture-independent analysis of microbiome. Why culture-independent? Why not doing it culture-dependent? What would you think? Would there be any disadvantage if we just avoid DNA sequencing and we just culture everything on Petri dishes? Looks like you have an idea. The microphone, please. Thank you.
You cannot control what is growing inside a bioreactor,
Yeah.
but I don't know if it's important to know and why. Maybe why is important. I don't know why it's important to know what's inside my bioreactor.
Mm-hmm. So the first part sounded really good about we don't know what's growing. So there are many unknown organisms. We just don't know who they are. They have never been sequenced. They have never been microbially characterized. So there are many organisms that we just don't know. So we can't compare them. And we also have the problem that many of the organisms just don't grow. We just don't know how to grow them. We can use all kinds of Petri dishes with media that we know, and they won't grow. So this made it almost impossible in the past to analyze microbiomes. And just since we came up with the possibility of DNA sequencing and other omics approaches, we suddenly have the possibility to take a look into an anaerobic microbiome. So therefore, these methods really were accelerating the whole process. And now it becomes much clearer how those microbiomes are composed of, how they are working, how they can influence ecosystem. So now we're really starting to work with them. And the first works, we have many works for now that are really descriptive. So very often people are describing it without really knowing how it is impacting the biogas plants. Because before we can make any assumptions about how to change the microbiome, we need to know what is there And this has been started several years ago, and now you can find hundreds of articles where people try to describe taxonomic profiles in anaerobic digester plants. Yeah, and step by step we are coming into a direction where we are thinking about how we actually could change a microbiome, how to alter its function, or how to make it maybe more efficient. But we just started with this, so it's approaching, but we are not done with this yet. So many people try to focus here on the core microbiome, on the most abundant microorganisms, because they are always there. And then we try to understand under which circumstances they change their abundancy. And a relatively new thing now is culture-omics. I told you that it's really difficult to culture anything from anaerobic digesters, but more and more technologies are developed which help also to automize culturing. And this goes into a direction where people say we could combine culture-omics with other approaches like high throughput approaches and omics-based methods in order to get a really holistic and integrated view into our anaerobic digester systems. Just to give you an example, this is not for microorganisms, this is for eukaryotic cells, but this is an automated system which can automate culturing for you. So you see here some tips and some tubes. Here's a robotic arm which can take these tips and can take some solutions from the tubes, then can put it on the dishes, the dish stocker. They are moved automatically in the CO2 incubator. So like this you can make kind of high throughput culturing in order to better study the microorganisms that we still don't know, or in this case, eukaryotes. Okay, now we talk a little bit about the capabilities of microbiomes. Why are they so interesting for us? What can they do? What do you think? Why are we so interested in microbiomes? Are there any specific characteristics which you would define for microbiomes?
For their capacity, so for the digestion capacity is the more important.
I didn't understand you. Can you repeat, please?
For their digestion process of microbiomes.
For the digestion process, yeah. But why? I mean, we have them in the digestion process, but why are we interested in them? What?
For degradation of organic matter.
Yeah. We can also do this with single cultures.
Yes.
Can we?
They need low energy.
Who was saying this? Oh, they need low energy.
To work.
No, I don't know. If I think about a single culture, I wouldn't say that a single culture is is less energy efficient in degrading it. Yes?
I think it's just to get more profit from them.
More profit in which regard?
To create more methane gas, to get energy. That's
So it's about efficiency.
yes.
Yeah, that's a good point. You also had something? I think the microphone is not working. Maybe there's a button.
I was wondering if maybe one of the most capabilities was treatment of water, maybe, and a process of digestion
Yeah.
to create methane,
Okay.
as my partner said.
Yeah. Okay, but this is already the application later on. But what's
Yeah.
the concrete function why we're so interested in them?
Oh, I don't know.
Okay. I think you had already a really good point. So the efficiency really increases because if you have just one single organism, it's just not capable of applying so many pathways. And if you have thousands of microorganisms, they are just combining incredible amounts of pathways in order to have a really efficient degradation. There are more things that are interesting about microbiomes, and now we will go through them step by step. There was an interesting article which was relatively recent from 2020. It was published in Cell Systems, which is a really good journal. And they talked about memory. It's strange because usually we wouldn't say that a microorganism has a consciousness. So this sounds strange. But they saw that they were capable of storing information, and this reminded them of a memory-like function. So of course this sounds really nice in the title. What did they really see? Why did they came up with this title? So it was an experiment with one kind of cells, and they have these pores where some ions can go out and come in. That's actually the same mechanism how our neurons are working. So we have neuronal cells, and then they have some fibers going out, and then we have these pores which opening and closing in order to transport an electrical signal along the neuron. And apparently they are using similar mechanisms. And then they did an experiment with light. So they had a biofilm. And then they were applying a beam of light on this biofilm. And then suddenly this pore was opening. So it was reacting to the light, but just in the spot where they applied the light. So they were waiting for some time, and then they were checking on the spot again. And then it was remembering. So it was still keeping the pore open in order to be able to react when the light comes again. So it was kind of remembering that this was happening. And imagine how complex this will be if we have a microbiome, where we have thousands of different species. All of them are remembering with strange mechanism certain information in order to act on certain selection pressures. This might be one reason why it's really difficult to predict the behavior of anaerobic microbiomes. That's one thing. Microbiomes are usually very difficult to predict in their behavior. And if we check on a biogas plant, we see some common things. But if we go to a really specific level, to the species level, we could say that almost every biogas plant is unique, because the things that are happening inside are just so complex. We also have a high diversity. That's something that I mentioned already. This was one of the pictures that you have seen already from my PhD thesis several years ago. And you see that there are quite some phenotypes that just look different from each other. I mean, we don't know who they are. That's what we already mentioned yesterday. But we see that there are many different ones. And there are not just many different ones. Also in the total number, there are a lot of them. So I was counting them actually. Not one by one, but I was estimating the number of them, and we have roughly 10 to the power of 10 prokaryotes per milliliter of anaerobic digester sludge. Let's compare this to the number of people that are living in Germany. So we have 82 million residents in Germany, and in 1 milliliter of sludge, we have 10 billion of prokaryotes. And they could all work for us. We just have to learn how. At the moment, they are basically doing what they want, and this usually results in the formation of methane. You had a question?
Yeah, I have a question. How do you do that? It's really complicated to count microorganisms in some one part. If you have many parts, how do you do that?
Yeah, you take a very small amount of sludge, you put it on a slide under the microscope, then we apply the DAPI staining that we learned yesterday. Then I can use software which automatically counts all the organisms in this picture, and then I just multiply the volume up to 1 milliliter, and then I have the final number. So it's an estimation. I'm not counting everyone. Yeah. Yeah, so there are many, and they can do a lot of work. And it would be nice if we actually find a way to produce something else than just methane, because methane is relatively cheap. I mean, it's better than nothing. It's better than just throwing it outside and then it dissipates into the atmosphere. But if we find a possibility to produce other metabolites, like different fatty acids, this could be really valuable. And that's what people are really trying to achieve, but at the moment, just on the level of publications, and we hope that eventually this will also come into the industry. So, I told you already how complex the thing becomes based on this memory function. So there are so many different microorganisms with such a high diversity, all of them are probably storing different information on how to react on different selection pressures. And now they are also interacting with each other. This makes it even more complex. And the interaction is quite interesting. So they can exchange some chemicals, they can be bound to each other, but then they can also make these kind of connections, which we also saw yesterday. And we heard that some of these connections are functioning like nanowires, like electrical wires produced from organic material on a nanoscopic scale. And then they use these fibers in order to exchange electrons. Maybe we can say it's the microbial internet or something. So then we have the interaction between different microorganisms on different levels of communication, which makes it again more complicated. So what you are seeing here, or what you're not seeing here because it's very small, is a map or is a list of quorum sensing. I don't know whether you have heard of quorum sensing before. It's the ability of microorganisms to sense other microorganisms. So they can kind of feel whether other microorganisms are nearby in order to react. So if there are too many, they can release some kind of toxin, or they can let them know that there is some food nearby so that they are coming and starting to grow. So they have some nice communication skills, and we are just approaching the tip of the iceberg. This is what we know so far. You don't have to read it, we are not going into the detail of it, but every point which you're seeing here is a different chemical or a different signaling molecule which these microorganisms are using to tell themselves how many they are in order to react accordingly. And yeah, this was downloaded from the KEK database. This is a really interesting tool which might be also helpful for you in the future. It's also free. And it's a database created in Japan. It's the so-called Kyoto Encyclopedia of Genes and Genomes. And they have the mission to collect basically all pathways that we know. And you can just go there and you have a searching field like in Google, and you can type in methane and then you get a total pathway for methanogenesis. Or you can type in quorum sensing and then you get this picture. And then you could actually click on each of these points to get a really detailed information from each of these communication skills. Yeah, but we are not going into detail of this because I mainly want to highlight the complexity of microbiomes, and I'm not going to explain you the deepness of molecular mechanisms behind quorum sensing. They also have quite strange forms of growth. So I told you yesterday that they're able to produce this extracellular matrix in order to build kind of their own home. So they're building something in order to be embedded in this matrix, and this allows them to fix their position at a place where a lot of food is present, or they could fix their position close to other microorganisms which they need to collaborate. So how to analyze this? This is really difficult because we can see this under the electron microscope, but then again, we don't know who they are. We can sequence everything, then we know who is there, but we don't know where they are. So this is really difficult. We might apply fluorescence microscopy again with the fish probes, the fluorescent in situ hybridization that we discussed yesterday. And here we could look for some very specific microorganisms. Again, it will be just a few because we can't apply so many different fluorescent probes, but we could maybe check the occurrence of 3, 4, or 5 different kinds of microorganisms to see whether they assemble together or whether they are farther away from each other. But this is really limited, so it would be nice to, let's say, more automate the whole process. And here's a trial to achieve this. This is an article which was published 2015 from my PhD father. And it's quite comprehensive on the mathematical level. So I think on the mathematical level, as engineers, you're much more trained than me. So I think I'm not the right person to explain this to you. I will explain it really simple. So here's some information how they were calculating it. You can read it later if you want. I'm not going into the mathematical details, but what they were doing was kind of modeling based on the Lotka-Volterra model. You may have heard of this Lotka-Volterra model, or at least you may know this model from the fox and the rabbit. So when we have a lot of rabbits, then the fox can eat the rabbits and the foxes will grow. And when the foxes eat all the rabbits are fewer, and then the foxes will die again. So we have like a shifted rising and decreasing of foxes and rabbits. And something similar could be on the microbial level. And for this, the authors here were just observing several time points based on sequencing. And then they knew at which time which organism was there in which abundance. And then you can make like a line when they grow and when they die. And then you can relate these lines to each other. And then you can postulate whether they are somehow dependent on each other or not. And this goes so far that the authors were even able to predict direct connections between these organisms. And in this scheme, in this diagram, which is based on this modeling, you see different spheres. And the bigger the sphere is, the more abundant this organism is in general. And then you see that the spheres are sometimes close to each other or further away from each other. And when they are really close to each other, then they are declared as one community. It's a community of microorganisms that likes to work together. And like this, we can hear several communities. So this is one way to approach this. There are other mathematical ways which goes really deep into bioinformatics, but I think the problem is not fully solved yet. It's still really difficult to really find out which microorganism is where and how they are related to other ones. So now, from this picture, we know that there are communities. We heard before that microorganisms can interact with each other, but now we have communities and they could also interact with each other. So this brings it again to a new level of complexity. And this idea was already catched up in a publication in 2014 by Davy and O'Toole. And they started with sugars that they wanted to ferment. And then they have several communities of microorganisms. All of them are involved in fermentation of sugar in order to produce organic acids. Sometimes you have diverse groups, sometimes you have really homogeneous groups. And then he also highlighted the different levels of communication, like intracellular signaling between the same kind of cells, or intracellular signaling between different kinds of cells. And then they can also have metabolic interactions. I told you that, or you came up with the idea that the efficiency is increased due to microbiomes, so they can supplement the pathways for others that are not able to perform these pathways. And then here we have an inner circle, which is just there to eat the organic acids, which are then converted into hydrogen. So this makes it again more complicated, because now we have all these levels of communication and interaction restricted to several levels of degradation. So you see, the deeper we are going into this, the more difficult it is to oversee what's really happening and to recognize clear patterns. Another problem is that here are apparently many communities, and all of them are fermenting sugars to organic acids. So this makes it really difficult to make like a functional scheme, like this microorganism makes this one makes this, and then this one makes this. No, there are like maybe 100 who are doing the same thing. This again makes it difficult to see clear and regular patterns in anaerobic microbiomes. This makes it really difficult for us to make any clear predictions how the microbiome will behave. But on the other hand, it leads to a really high robustness. And next to the efficiency, which you mentioned, the robustness is something really nice, because in a biogas plant, there's little we have to do. We basically throw in our sludge together with the waste, and then the microorganisms organize themselves based on these complicated interaction patterns in order to produce the methane. And it's very difficult to disturb this process. If we want to produce methane, this is nice. But if we want to change the microbiome in order to produce a very specific metabolite, this really becomes a problem. But in order to change the microbiome, we really have to understand how it works. And at the moment, this is quite difficult. Okay, talking about different possibilities to change the microbiome. This brings us a little bit into medical science. I guess all of you have been already sick in the stomach. And if you're sick in the stomach, then your microbiome is altered. Something changes in the composition of your microbiome. And of course, it would be nice to repair this somehow. Very often this happens automatically, but not always. So if everything is okay, if the composition of microorganisms is in the right composition, then we have optimum health, we have a high fitness, fast growth, high productivity, and high fertility. But then when it's disturbed, we're getting sick, the fitness gets lower, slow growth, low productivity, low fertility. And well, that's something that medical science tries to change based on microbiome engineering. I thought that something is missing, or at least if we're talking about biorefinery or anaerobic digestion, because I think it would be nice to directly alter the microbiome somehow in order to come to something which is maybe better than this homeostasis. We don't know for sure whether this is possible, but we have some good ideas. We will come back to this later. And I just want to highlight some possibilities to change the microbiome. Many of these possibilities are already applied in medical sciences, but I could imagine that some of them will be applied in the future in order to specifically change a microbiome to alter its function. I think you all have heard about prebiotics and probiotics. So prebiotic is something that we can eat, which is not really degraded, but then it somehow changes how microorganisms act, so influences the microbial community. And we can also use living organisms. I suppose you all know this Bifidobacterium bifidum. It's very often in yogurt, and you can eat it to strengthen your microbiome. We can also perform a microbiome transfer. And this actually happens already because people which are really sick can swallow pills which contain the fecals of really healthy people. Of course, this is kind of disgusting, but if you're really sick, then maybe you don't care if it helps in the end. So this is a nice way to heal a microbiome. And this is something that we actually apply already in biogas plants. So if a biogas plant is not working very well, then we go to another one and we just take the microbiome from this plant and then we bring it to the sick one. We can also use signal molecules. I was asking myself what's the difference between signal molecules and probiotics. I would say that the signal molecules maybe can be degraded and the probiotics are usually not degraded, but I haven't checked on this. We also have medication like antibiotics, antimicrobial peptides. And this is actually affecting already biogas plants because very often we have antibiotics in the microbiome. For example, if we use the manure from cows, very often animals are treated with antibiotics, and then these antibiotics are entering in our biogas plant and could disturb the process. And we can just change the outside. We can change the parameters under which the microorganisms are living, like the pH value, the temperature, the substrate composition. We can add certain nutrients, so we can just change the way of life for the microorganisms, and in return, the microbiome will also adapt. And then the last option is synthetic biology. We can actually construct microbes. This is already possible. There are many groups which can do this without problems. Can also use viruses. So would this be helpful for a biogas plant? Would you think it's a good idea to specifically construct a very efficient bacterium that we are throwing into our biogas plant? You are nodding. Why? Okay, so you say we make them more efficient, put them inside, why not? Okay, other opinions? Yes?
Maybe no, because microorganisms need times to adapting to that system or some other microbiomes.
Yeah, that's right. So I agree with you. It would be really nice to have efficient microorganisms. We can build them, but then if we throw them into the biogas plant, they will possibly be degraded. And we could throw in every day tons of microorganisms, but this is in the end not economic. So at the moment, we have no good way to specifically construct microorganisms for the biogas plants, but maybe this comes eventually. Maybe one day we find a good selection pressure and we can program these cells in such a way that they can survive the selection pressure, and then we can achieve conditions which allow the enrichment of a specific organism. But that's still very far away. We are not there yet. Okay, I would like to talk a little bit about antibiotics. I told you that they are already in anaerobic digesters because we are treating the cows or other animals with antibiotics. This is a problem, not only in biogas plants, but everywhere. And that's something that also is of high importance for you as engineers. So what are antibiotics? Antibiotics are a group of therapeutics. There are other therapeutics like antiprotozoals, antifungals, antivirals, anthelmintics. What are anthelmintics? Did anyone try them before? Yes?
Is medicine against worms? Like
Yes, that's right. That's absolutely right. So originally, these therapeutics, or more specifically the antibiotics, were produced by fungi, also by bacteria, but basically by fungi to kill bacteria. And here are some examples. So we discovered them relatively late. The first one was in 1910 by Paul Ehrlich, and he discovered the salvarsan, which works against the syphilis. Then we also have the penicillin. I guess you all have heard about Alexander Fleming. I guess you also heard about chloramphenicol, aureomycin, tetracycline. Those are typical antibiotics that you can take if you're sick. So here's a historical timeline on the discovery of antibiotics. We are not going to read all of them. I'm not so interested in all the names, but this is really important. So somewhere in the 50s and in the 60s, we discovered a lot of them. And I think it's logical. So we discovered the first of them, then we saw, well, this can heal us, so we should search for more of them. They searched and they found more of them. But then eventually this came to a stop. And now we keep doing research, but we are finding not so many anymore. Some of them were discovered here, and nowadays millions are invested to, billions to find new antibiotics. Why is this so important? Why are we doing this? Why not just staying with the first one? Yeah?
Because bacteria develops kind of resistance
Yes.
against the first one.
Yes, that's right. They adapt. That's a problem. Apparently, they adapt much faster than we can find new ones. And this is already a problem. You all have heard about the multi-resistant bacteria that we can't treat anymore properly. So this is already a time where people start to die because of simple sicknesses that we could treat easily before with antibiotics. So how is this relevant for you? I mean, very likely you're not going to search for new antibiotics. How is this relevant for an engineer?
Maybe because our health.
For your health?
Yeah, for my health.
Well, this would be a personal reason.
I need to be healthy.
But I mean, if you think about you as an engineer,
That we have the job to maybe about the water, to clean the water. So
Yes.
if we can kill them, we can clean the water.
that's right.
properly.
So this is quite a hot topic at the moment in Germany. We are talking about the fourth cleaning stage, because after cleaning the water, we removed all the biomass and the water looks quite clear, but then sometimes the antibiotics are not degraded still inside. So how does an antibiotic work? How much time do we have?
Excuse me?
Okay, 1 hour.
Sorry,
So-
I have a question.
Yes, please.
What is the most common antibiotic that you find in Germany? Because
The
here in Mexico,
most
it's
common
really-
one.
Yeah, what is the most common?
I don't know. I mean, I didn't watch a statistic about this. I mean, there are some that I mentioned before, like this chloramphenicol, auromycin, tetracycline. Those ones are used quite often. But I mean, it depends on the microorganisms, because if you have a microorganism which is already adapted, then they just have to try another antibiotic. I once had an bacterium. I was sick, and they were analyzing it, and they said, well, there's just one antibiotic that worked. So I think this concerns everyone already, and it's quite probable to get such organisms. I mean, in many cases, our immune system will protect us, but then especially for old people who can't defend properly against simple sicknesses, then for them it's a real problem. Yeah, and I think it's also a big problem that people take antibiotics too easily. So if you're sick, many people just buy antibiotics and take them, and very often they are just not working because they have a viral sickness and not a bacterial sickness. And antibiotics are not working against viruses. So, but people just buy antibiotics when they are sick, and then they're spreading. So how does an antibiotic actually work? So this is the case of penicillin. Here we have a complicated name, the D-alanine transpeptidase. You don't have to learn this name. But still, to explain it to you, alanine is an amino acid. Transpeptidase is an enzyme which is able to transfer these amino acids on other proteins. And this is really important to build the cell wall of bacteria. So this penicillin goes into this enzyme. There is this induced fit theory, or K-log principle. You may have heard of it. So the antibiotic goes into the active center of the enzyme, and usually in this active center goes then our alanine in order to be transferred on the structure, on the cell wall. Well, but if it's blocked by our penicillin, then this can't work anymore. So this is not a specific scheme for the D-alanine transpeptidase. I just was looking for a picture that shows the K-log principle. In this case, here you have a substrate which goes in the active binding site, and then there it's splitted, and then you have two products. This could be a possible reaction of an enzyme. Yeah, and the penicillin is now blocking this site for the alanine transpeptidase, and after this the bacteria can't divide because they can't grow the cell wall anymore. So then they will just die. There are other mechanisms. I'm not going too much into detail of them. I mean, we just talked about the penicillin, we have the arsphenamine, and this is toxic due to heavy metals. Heavy metals are also quite interesting for you. Why are heavy metals so toxic? What do you think? I mean, we all know that they are toxic, but why? What makes heavy metals toxic? Why are they toxic for us? Concentration, yeah, but why? Pardon?
They don't have a metabolism function.
Well, but then we could just ignore them. I mean, in the moment when they are entering our metabolism, they are really destroying us. Why is that? You have an idea? Yes?
Because heavy metals is not possible degraded by any biological or chemistry process.
Yes, this is right. We can't degrade them, so we can't get rid of them. but still we don't know why they are killing us. You have an idea? Yes?
Yes, the heavy metals can act like as a nutrient, but doesn't have the same function for that nutrient, and the enzymes or the proteins doesn't work well, and that make us sick.
Yeah, I haven't heard about this, but could be. So you mean that they are working like in the active center of an enzyme, but then it's the wrong metal, because sometimes we also have metals in proteins, and then the functionality is not right anymore.
Yes.
Yeah, this could be. But there's another reason. Yes.
I have read something about the warning in the brain, in our brain, or the human brain.
Yeah.
But for example, I don't know, aluminium.
Yeah. Okay.
How do you say aluminium? But the main, yeah, I have read about something like that. For example, is the warning, the brain, the human brain.
Yeah. I mean, if we have a deodorant, some claim we shouldn't have aluminium inside. So it's already acknowledged that aluminium could be toxic. But then again, the question is why?
Or, for example, the central system human.
Yeah, but what does it do? How does it change? I mean, it could be everywhere in our body. It could be in the brain, it could be in the central nervous system, it could be on our skin. Everywhere where we have the heavy metals, they will destroy us. Why?
I don't know exactly, but I have read something like this.
Okay.
I read that some metals are used in waste, in purification, like silver or copper, and I read that it is used because these metals goes into the cells, and I can explain it.
Yeah.
But it goes right into the DNA, and the cells don't have the capacity to reproduce. And if they don't reproduce, after some time, they die.
Okay, but why?
They replace the other elements.
Well, this goes already in a good direction, yeah? Can anyone specify this more? Okay, then I will resolve it. I guess you all know that heavy metals are catalyzers. You can use them to catalyze something. They are extremely reactive. And why are they so reactive? Because they're really huge atoms. They have quite some electron shells, and they have an immense cloud of electrons surrounding them. And then we have these quantum effects of fluctuations, and they are much stronger if you have many electrons, so the possibility increases that just one electron jumps off its shell. And if an electron jumps off its shell, then it will react somewhere. And then once this electron left, the metal will fill the space again, so it's trying to get the electron from somewhere. So there's a rapid exchange of electrons with everything surrounding the heavy metal. This makes them extremely reactive, but this also makes them a good catalysator. But it would kill us then. Well, this was used for the azethamine, and of course we are not using this anymore because we can't, the heavy metal is not just killing the bacteria but also us, so it's not the best option. But then here's another one, like the chloramphenicol. It works again similar like this penicillin, blocking some kind of enzyme. In this case, it's the translational apparatus with the ribosomes where we are building our proteins. You know this already. And the aureomycin has a similar mechanism. It's also disturbing translation. It's disturbing the aminoacyl tRNA. This is the protein which is carrying the amino acids to the growing protein in the ribosome. Okay, how do the bacteria actually learn to be resistant to a certain antibiotic? So here's a picture which shows this pretty well. We have two bacteria, they are quite close to each other. Here you see the DNA, and here you see something that we call a plasmid. And they are able to make small messages out of their DNA. They can wrap them into a small plasmid, and then they can send them like emails to another cell. So they can make a conjunction here, and then the plasmid just enters in the other cell, and this plasmid could wear this information for the resistance against this antibioticum, and then the other cell is also resistant. So there's an interesting article, Baquero et al. From 2008, and they were talking about a kind of environmental reactors. So here you see different frames. Each of this frame is regarded as an environmental reactor by them. I also give you some explanations, but I will just explain them to you like this, and you can read them later on in order to re-understand what I just explained here. So we have here the first reactor where antibiotics come into use by animals or by humans. Here we have microorganisms which are growing on animals or human microbiota, and they could now be carried away by the animals, by the humans, into farms or aquaculture or hospitals. So when I'm sick, I'm going to the hospital, I get the antibiotic, and some of the bacteria on me might jump off me in the hospital with the resistance against the antibiotics. From the hospitals or from the farms and aquaculture, they could jump into wastewater or sewage treatment plants. Here's another color. Those are also microorganisms, but environmental ones. So those are not the ones which are specifically go on animals or humans. They just go everywhere in the environment, into the air, into the water, into the soil. And there they are carrying their resistance around. So from the environment, they can go back to the human and tell bacteria on human how to be resistant against antibiotics. Yeah, and the further we go, the more environmental bacteria are catching up this information in wastewater effluents or sewage treatment plants, or then even in surface groundwater, soil sediments. And in the end, this information, how to survive these antibiotics, is everywhere and goes from everywhere back to the humans, so that the pathogens that are sitting in us learn how to survive these antibiotics. Well, as you said already, as an engineer, for you this is important in order to find ways to destroy all antibiotics in the wastewater so that they not reach the remaining environment. And I think this could be much more successful than finding new antibiotics, because so far this goes quite slow. Maybe there comes a new technology or something which accelerates this, but so far I don't know this. I haven't seen this. So I just see that it's more and more difficult to find new antibiotics, but we can really find efficient ways to destroy antibiotics, and that's something that you could do in the future in your field of work. Okay, so at this point I will summarize what we have learned about microbiomes. So We have learned the importance of microbiomes in order to act as a really complex system with high efficiency. We heard about the importance of cultivation-independent approaches in order to see what's happening at all in a microbiome, or at least to see which microorganisms are there. We saw several communication levels. So we have several microorganisms that can engage with each other. We have communities that can engage with each other. They have several signaling ways. Then we have this chrome sensing, for example, prebiotics, and so on. And we have heard that we have a really high diversity, which makes microbiomes really strong and competitive, which makes it easy to produce methane, but which makes it really difficult to produce anything else. Okay, are there any questions so far? Everything clear? Everything understood? Okay, great. Then we will come back to the metabolism. So now you have to think a little bit more about biochemistry. I will try to make it simple, but we will now talk again about the electron transport chain, or at least at some part in this subchapter. So I hope that you will remember something from the electron transport chain yesterday in the photosynthesis. So, and I would like to talk about a functional classification of microbiomes, because we learned now that microbiomes are so complicated that it's really difficult to see any clear patterns in the microbiome. But still, we think that we can distinguish microbiomes based on the presence of oxygen. So depending on whether oxygen is there or not, a microbiome can behave really differently. Do you know why? What is the importance of oxygen for the metabolism? Why do we breathe?
As an electron receptor?
As an electron receptor, yes, that's quite good. Any other opinion? You were raising your hand?
I think it's the last receptor.
Yes, right. Great. And you? The same, okay. 3 similar answers, then it must be true. So, it's an electron receptor. But do all organisms need this electron acceptor? No. What are the microorganisms in biogas plants doing?
Oxygen is toxic for them, and that kind of microorganisms.
Yes.
But not for everyone needs oxygen for.
Yeah, for some it's toxic, that's right.
The process, no?
Yes, that's right.
I understand that. Yeah.
Okay, but what do they do when they have no oxygen as electron acceptor? I mean, we heard in the beginning of the first talk, I think, that organisms also have to get rid of electrons. So at some point they can't gain any energy anymore from the electrons. They try to get rid of them. This could be happen with oxygen, with the reaction of oxygen as final reaction to produce water. But what do microorganisms without oxygen do?
They use organic compounds?
Yes, that's right. So they use organic compounds, but usually they are much less efficient than the oxygen. So they can generate much less energy than oxic organisms. So here we're speaking again about the ATP. I hope you all remembered the ATP. It will not be the last time that we're talking about ATP. So if we have oxygen present, then it's much easier to produce the ATP. Here again, the reaction of the ATP, we have seen it before in the photosynthesis. we have the adenosine triphosphate, and the adenosine triphosphate can release one phosphate in a chemical reaction, and this in turn releases some energy which can then push any chemical reaction inside the cell. So here another pathway. You don't have to learn it. Maybe you have seen it before. This is how we are degrading sugar. I mean we, as humans, but also bacteria, basically all kind of organisms that are eating sugar. Well, there is an alternative pathway, but this one is really often found. This is the main pathway to ferment sugar and to produce energy of sugar. And you see here several steps. You don't have to remember them all. It would be nice if you know the basic functionality of this so-called glycolysis. So first we have a reaction with ATP, which might be strange because we actually want to produce ATP, and here we are losing ATP. And then already 2 molecules of ATP just for 1 molecule of glucose. We end up with fructose 1.6 phosphate. Well, with this ATP that are reacting with the glucose, we have a really active molecule. So the more energy we put in, the more reactive the molecule becomes. And now it suddenly splits. With this high reactivity, the molecule can be split into shorter molecules, which then have just 3 C atoms. Here we are still at an hexamer with 6 C atoms, and here we have now 3. Then we have here an isomerization. The dihydroxyacetone phosphate also turns in glycerolaldehyde 3-phosphate. So we have 2 similar molecules, and they now react again with phosphate, but not from the ATP. They are so reactive they can already take up normal phosphate, which is swimming around, and then suddenly we can yield energy from this. Now they can recharge other ADPs. The first one, then after a few transformation steps, a second one, and if we sum it up, then we have here 4 ATPs. We have consumed 2. So in total, we are producing 2 ATPs from the glycolysis. Is this a lot? Would you say this is a lot? I don't know whether you have heard about the glycolysis before. You are nodding. Okay. Yeah, so no, it's not a lot. It's very little, actually. But we can do something else now, because we still have this pyruvate. It's like a last metabolite, which now goes into a second pathway, the so-called citric acid cycle. Maybe you have heard of this before. Again, you don't have to learn this pathway, but it would be nice if you remember the basic function of it. There are two metabolites that you may remember, actually. Which are these? I will ask more specific. Does anyone remember the oxalacetate? I think you have it. No? From where do we remember the oxalacetate? We have seen it before, but where? Yeah, right. And where? What is it doing in photosynthesis?
It catches CO2.
Yes, perfect. It's for the C4 metabolism. That's right. And then we have another one, the malate. What's the malate for?
It stabilizes.
Yeah, kind of stabilization for what?
Yeah. To the CAM plants.
Yeah, right. to store it for a longer time, for this time separation in photosynthesis. Perfect. Okay, we know these two, and the rest you can forget. But the important thing is that we have here a circular way, and after this circle, when the acetyl-CoA, so the acetyl-CoA is formed from the pyruvate, and the acetyl-CoA now goes through this circle, for the circular pathway, and then you see many things which are coming out. Basically CO2, and then hydrogen, which is attached to this NAD+. I guess you still remember the NAD+ from the photosynthesis. So, but we are not plants. So what are we doing now with the NAD+ or the NADH? It's difficult. We haven't talked about this before, but maybe you have a creative idea. You look like you know something.
No, can you repeat the question, please?
Yeah, I asked why do we have so many NADH pluses? So all this this residue from the pyruvate is now transformed into basically CO2 and hydrogen in the form of NADH+. So we heard about it in regard to plants, in regard to photosynthesis, to build up biomass, but we are not plants, so we will not build up biomass from this, or probably not. So what else could we do with this? Any suggestion? Any idea?
Well, that we use then with the electron change to create ATP.
Yes, that's right. That is a good idea. So actually we are kind of reversing the flow in the photosynthesis. We are trying to use the electron transport chain to produce NADH, but here we actually want to get rid of it. So we are using the NADH to build up a gradient of protons, and we release the electrons into the electron transport chain in order to produce ATP. So we basically use it for energy production. If we're doing this, we're getting all these products. There's one GTP. I mean, we know what to do from ATP. We know what to do with NADH. We have the FADH, which is very similar to the NADH in its function. But then we also produce here GTP. What is this? For what do we need a GTP? Any suggestions? Have we seen this before? You're shaking your head? Not seen before, okay. Anyone else seen it before? Well, it's a nucleotide, like the adenosine triphosphate. We also can have guanine triphosphate. It's also very energetic, but usually we use ATP as chemical form of energy in the cells. But then you may know that the nucleotides are also important for our DNA. So it's going to be built into our DNA in the end. So if we sum up all this, so if we make the calculations, how much ATP could actually be produced from this NADH and FADH, if it goes to the electron transport chain, it accumulates to 36 ATPs. But the electron transport chain at least as we use it, can only be powered with oxygen. It's the final electron acceptor, as you all knew already. So only if we have oxygen, we can produce this, where was it? This 33 ATPs. And if we have no oxygen, then we end up with these two, where was it? With this 2 ATP from glycolysis. This means 95% of the energy remains unused if we are not using oxygen. So what does this mean? It means that organisms which are not able to use oxygen will grow much slower. They will produce less heat. They are just less efficient than organisms which are using oxygen. And this makes a huge difference because in wastewater treatment, at least in the sludge activation, we are having an oxic stage which is extremely fast. It allows us to clean the water very fast, but basically all the energy is lost and dissipates into the atmosphere. But we still do it because we want to have it fast. In contrast with the anaerobic process, where we have no oxygen present, there's so little energy, and since there's no final electron acceptor in the form of oxygen, they're not able to convert everything in CO2. And that's the reason that we actually can produce something what we can use later on. Okay, so to summarize this, to summarize the electron transport chain, or to show it, I haven't explained it in detail, but it's very similar to the photosynthesis. I have a short video which shows how the electron transport chain works in order to produce ATP with oxygen. So we should start it now. No? Can you please start it? No? Is there someone there who can start it? I will wait a second. Maybe he comes back or so. Otherwise, we have to proceed, and you can watch it later on YouTube. Now something happens.
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH. These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force. During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed. The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP. This enzyme allows protons to pass back into the cell and couples the energy released in this process to the phosphorylation of ADP to form ATP.
So, I think this is really easy to understand once you understood the photosynthesis. So, the process is very similar. So is it clear, or do you more or less understood it? It's clear. Perfect. Okay, so there's an interesting question. So is the citric acid cycle important for anaerobic organisms? Because actually we have no oxygen. So I was asking this question myself, and I was reading a little bit about it. And of course, the metabolism takes place, the pathway takes place, but without the electron transport chain. So they're producing all these NADH, and they have to get rid of it somehow in a different way. But still, this wheel is turning, and it's like the central wheel of metabolism. So many things are connected to this citric acid cycle. And a really important thing here is the supply of important precursors, like, for example, the malate and the oxalacetate, where we already have seen its importance for photosynthesis. So here you see how these pathways are interconnected, and therefore the pathway exists, but not for the production of ATP based on this electron transport chain. Okay, we can also put this in numbers, because the oxygen has an oxidative power which we can measure. And I was looking for articles which measured the oxidation potential in anaerobic systems and oxic systems. Here you see an oxic system, an aerated tank of wastewater. There we reach up to 420 millivolts. And then in an anaerobic system, we have minus 450 millivolts. It can be a bit different depending on the system, but I think these numbers are roughly quite okay. And if we just measure the difference between them, yeah, we have almost one volt. So it's close to the voltage of a normal AA battery. This is just to show you how powerful the oxygen actually is, and I think it makes it a little bit easier to understand what actually is meant with oxidative power. So you can imagine what you can power with such a battery. And that's the power that life is using also to create energy. And as I told you, if we are now in this system with high oxidation power, then we can work better, we can work faster, but we can't produce valuable metabolites that are enriched in the anaerobic system. This is really important for engineering now. Maybe one of you or several of you are going to construct reactors eventually. And if you are working with an anaerobic reactor, you will have the problem that no biomass is enriched. So we can call this a flushing reactor. So you put in your biomass and it flushes through, but you will not really enrich lots of biomass, which is, it's a pity because we need a lot of biomass, a lot of microorganisms which are growing on the biomass. We would like to have these flocks in wastewater treatment to separate them from the water. So how can we achieve this under anaerobic conditions? There are ways, and maybe you have heard some of them already. So we have these flakes from water treatment, but for this we need some kind of activation process. So for example, we can have an activation in a normal water treatment plant, and then we have the flakes which are working in the anaerobic stage. This could be a possibility. We could also work with membranes. You could implement membranes into your reactor, and if you have small biomass molecules, they will be retained at the membrane, so they will not be flushed through, and like this you can enrich biomass at the membrane in order to increase the reaction velocity of your system, and to just increase the number of microorganisms that can be present here to work. We can also produce pellets, or yeah, I think granules might be a better term here. So if you have a so-called UASB process, this is also known as upstream anaerobic sludge blanket. Maybe you have heard of this. There you have a selection pressure based on an upflow. So you have your wastewater, it goes into the reactor, it's flowing up, and all the microorganisms will basically flush through. But then you might have some granules that are forming just by accident, and they are able to resist the upstream. So they will sink a little bit down, and when they are retained, then they can grow, they can become bigger. And due to this selection pressure of the resistance against the upflow, you are able actually to grow granules, which are then forming a kind of bed in your reactor. And this can be optimized in an EGSB reactor, where you have just a buffle at the top, which kind of reflects the biomass so that it can't flow out properly, and then you can even better produce these granules in your reactor. We can also use biocarriers. We can use solid materials that we are throwing into our reactor. It could be a fixed bed, it could be particles, could be plastic particles, whatever, basically every structure where microorganisms can grow in, and then they will grow there, will be fixed to the carriers and do their job there. And of course, we can combine all these methods to build a kind of hybrid reactor. So those are tricks in engineering to enrich the biomass in your reactors in order to make the microbial activity more intense for degradation processes. Okay, so this makes it really useful now to really distinguish between aerobic and anaerobic microbiomes. I tried to show this kind of scheme. So we have the aerobic microbiomes, we have the anaerobic microbiomes, and we can have something in between. I don't know whether you have heard before of microaerophilic microbiomes. What is a microaerophilic microbiome? How is it defined? How can we detect whether a microbiome is microaerophilic or not? What would you say? Any idea? No?
They are the microorganisms that could live with the presence of oxygen and without them.
Yeah, that's right. Those are facultative microorganisms. But when are we in a microaerophilic situation? How much oxygen is there? How can we measure this?
Is when you have the oxygen, the minimal oxygen to the metabolism of that microorganism.
Yeah, and how do you measure this?
You can measure that, no?
So you would measure it how?
Maybe you are adding some oxygen, but you can read it with a membrane or with a sensor.
So, okay, you say we have a sensor and we have some inflow of oxygen.
Yes.
But then how can we make sure that it's not aerobic? So you measure, where do you measure the oxygen?
With a sensor with membranes or with a light sensor? Or?
Yeah, let's say we have a really good working oxygen sensor. But where would you place the sensor?
water, you are measure the dissolved oxygen in water, but maybe the microorganisms are taking that oxygen that you are adding to the water.
Okay, I think that's a really good answer. So I was looking for an answer, and I found this 2 to 10% of oxygen. So 2 to 10% where? So it could be in the atmosphere, it could be in our biogas. But then I think we have a big error because the oxygen could react. I think you're right, it's already much more accurate if we measure it in the water itself, in the sludge itself. But then still we don't know how fast the oxygen is consumed. I mean, depending on the amount of microorganisms that we have, they can probably use the oxygen relatively fast. And if we don't have so high density of microbes, they will consume it not so fast. And maybe you have a leakage somewhere, maybe you have pipes which are not gas tight, and then all the time oxygen is entering in your reactor, but they consume it so fast that you don't measure it, not in the liquid and not in the air, not in the biogas phase. Is this microaerophilic or not? It's a difficult question. So when I read about microaerophilic conditions, people usually say that they apply some oxygen and then it's microaerophilic. But it's really difficult to define a clear border where it's microaerophilic. So are we already out of time? 3 minutes, so I can finish this slide. So I think it's really useful to define this kind of microbiomes, to say we have aerobic microbiomes, we can have anaerobic microbiomes, and we can have something in between, which is difficult to measure, but it still exists, and we use this term, microaerophilic microbiomes. The borders are fluent, but we try to draw them still, and a typical aerobic microbiome is, for example, our wastewater treatment with the sludge activation or the upper part of the soil. And we can also have the anaerobic microbiomes, typically with biogas or maybe biorefineries. And when we are in between, for example, the compost, then it could be a good example for microaerophilic microbiomes because they are continuously pending between aerobic and anaerobic conditions, and somewhere in between they are certainly also microaerophilic. And this has not only a big impact on the energy production for the organisms in respect to ATP or in respect to the enrichment of valuable metabolites like methane. It is also important in regard to the kind of organisms that are growing here, because under anaerobic conditions we have mainly bacteria and mainly archaea, sometimes fungi, and very little, almost no eukaryotes. But the eukaryotes are growing very well when we have an aerobic microbiome, which again allows us to produce the flux in the activated wastewater that we are needing to separate the water from the biomass later on. Okay, I will stop here because time is over. We will continue here tomorrow. And thanks for your attention. Do you have any questions? Anything that remained unclear? You can ask now. You can also ask tomorrow. Okay, then thank you very much and see you tomorrow.